and letting a stem cell transplanter crash the party. I've learned a lot these past few days about GU. So today I'm going to talk about CAR-NK cells and kind of what we've done at our institution in developing this cellular therapy. So cellular therapy in general has been pretty new to solid cancers, and NK cells in particular has been even newer. So here's my disclosures. So we know a lot about CAR T cells, but CAR-NK cells actually might provide a more advantageous cell type. Because these NK cells have low to absent GVHD, they can become an allogeneic product, which means they can be off the shelf, potentially low cost. And because we know that the immune cells from cancer patients are often very defective, we can use a healthy donor instead. Furthermore, from all the trials we have right now of NK cells, they've been shown to have very low toxicity, almost no cytokine release syndrome, neurotoxicity that require these CAR T cell drugs to be delivered in specialized facilities. And so these CAR-NK cells could potentially be given in the community setting, thus increasing their availability. Finally, CAR T cells depend on just the CAR for their to attack the cancer cells, while NK cells naturally have their own ability to attack cancer cells. And so we can use both NK cells' natural ability as well as the digital CAR therapy to kill the cancer cells. So fortunately, at MD Anderson, we have a cord blood bank, and so we can use these cells to generate our NK cells. And so at our institution, we performed the first ever phase 1/2 CAR-NK CD19 CAR-NK cells in patients with relapsed refractory B-cell malignancies. In this clinical trial, we use cord blood-derived NK cells. We transduce them with a retrovirus vector to express the CD19 CAR-NK, along with IL-15 to promote their persistence. And then we gave the patients lymphodepleting chemotherapy through therapy and cyclophosphamide, and then infused fresh CAR-NK cells on day 0. And so the results were initially published in the NEJM a few years back, and then the final report was just published this past year in Nature Medicine. And so for this trial, we treated 37 patients. And of these 37 patients, we did not identify any cytokine release syndrome or any neurotoxicity or graft versus host disease. And at day 100, we had overall response rates of almost 50%. And so we investigated this a little bit further, try to identify, why some patients responded and some patients did not. And what we found out was that not all cord blood was actually identical. So even though they're, quote, healthy donors, they're not actually all the same. And so for the investigation, we found that cord bloods with a nucleated RBC of less than 8 × 10⁷ and a collection to cryopreservation of less than 24 hours actually had a superior outcome. And you can see that in the PFS graph here. The orange line is from what we call the optimal cord, which had a much higher response rate than the patients who received the suboptimal cord. And we didn't know this before this trial. And we found that with further investigation, we found that these optimal cord bloods were very highly functional, were able to kill tumor multiple times in rechallenge studies in vitro, and were enriched in effector-related genes. In contrast, the cells from suboptimal cords, probably because they had higher nucleated RBCs, you can probably suspect that maybe they were from mothers that were more stressed. They had more hypoxia and just induction factors, cellular stress programs, and so they did not function as well. And so we took what we learned from that trial and we started to apply it to other tumor types. And the one I'm going to talk about today, right now, is going to be the TROP2 CAR-NK cells. So we're applying this now therapy from hemolignancies now into the solid cancer world. And so TROP2 is, of course, expressed in multiple tumor types, including a number of GU cancers. You can see bladder cancer is the second highest expressing in the CCGA plot. And of course, the first FDA target for TROP2 is sacituzumab, the antibody drug conjugate approved for breast cancer. It was originally approved for urothelial cancer, though it's been withdrawn since then. And so we initiated a phase 1 dose escalation expansion trial for TROP2 CAR-NK cells in patients with advanced solid cancers. And so this time we're using the optimal core blood units. We're isolating these NK cells. We're transducing and expanding them with TROP2. And then another twist, while the previous study we had to use fresh NK cells, we've finally been able to crack the problem and able to freeze these CAR-NK cells and thus actually develop and bank these CAR-NK cells so they can be truly off the shelf. So patients will receive lymphodepleting chemotherapy, fludarabine cyclophosphamide, and then day 0 they'll be infused with the TROP2 CAR-NK cells, and that's pretty much it. Very simple treatment. This is run by the PI is Dr. Dumbrava in the Department of Investigational Cancer Therapeutics in conjunction with the stem cell department. Of course, all this work is done on large number of people. And MD just announced the Institute for Cell Therapy, which is headed by Katie Rizvani, of which I'm, all of us are part of the institute. And our goal is to bring more cell therapy to cancers, to treat cancer patients much faster and more rapidly. So thank you.